So we're going to get started right away just talking about vision and how our eyeballs and our brain actually engage in that particular sensation, right? Remember, sensation being our actual perception of a stimulus. And in this case, vision is when we encounter waves of electromagnetic radiation. We know that our eyes respond to some of these waves, but not all of them. So if you look like at this image here, right, I'm sure in science at some point in your lives you've seen these different types of waves, gamma rays, X-rays, all the way up to AC circuits. Our eyes are able to see the light waves from just a very minute part of this kind of continuum. And our brain is able to turn these energy wave sensations into colors. So basically, this is how we see in color vision, right? We see light waves that are different in their complexities in terms of their, as we're going to see here, in terms of their frequency, in terms of their amplitudes, right? How high they are, how often the wave, the crest and the trough, is that what it is? Science, am I right? How often that takes place. And this is how we perceive color. And then ultimately we recognize like images and different components of our visual field by how light is bouncing off of those images, right? And that's how our brain is taking into account. Right now I'm looking at my kitchen. I see that I have butcher block countertops and like a dark teal greenish bottom counter with white tops, right? Like I'm able to perceive that because of the light bouncing off of those surfaces and my eye interpreting the different wavelengths or frequencies as well as their height or amplitude, right? So here, looking at these images, we perceive the wavelength or frequency of the electromagnetic waves as color, right? So what color are we looking at? Shorter wavelengths have a bluer color, longer wavelengths have a reddish color. And then the second piece to this is our perception of height and amplitude as intensity or brightness. So like how bright a color is, how bold it is versus how like dull and light it might be. So anything that has higher amplitude, it's going to be more poignant, it's going to stick out more, and anything with smaller amplitudes, it's going to be a little more dull. So what does the eye really look like? Here is a diagram of the eye. You'll notice in your chapter 6 terms list in terms of like what you actually need to know. You really only need to be able to like define our rods, cones, retina, and fovea. That doesn't mean in order to have an understanding of those four pieces of the eye, the rods and the cones, which aren't on here, the fovea and the retina, in order to have an understanding of what those do, we have to really get a bigger picture of what everything does. So looking at this particular image, you'll see on the left, kind of by the yellow title box, there's a candle, right? And so we know light passes from the candle and passes through the cornea and the pupil. Gets focused and inverted by the lens, inverted meaning it gets flipped upside down. Everything that our brain takes in is actually upside down and our brain flips it right side up. And then the light lands on the retina where it begins the process of transduction into neural impulses to be sent through the optic nerve, right? So this is all like, it's crazy what our brains are doing like 24/7 with just like looking around and looking at things. Like this is so fast that this light is coming in, like I said, through the cornea and pupil, focusing, it's getting inverted by the lens, it's hitting back on the retina where it starts to be converted into neural impulses, which then goes through the optic nerve and is sent right into our brain, into our occipital lobe, where it is actually turned into a perception of like, what does this mean? What is this stimulus? What is it? What am I looking at, right? And so this is a very, very obviously very fast process. It's happening consistently, continuously throughout our daily lives, and we don't even really know it's happening, right? We also know when we're looking at this image of the eye, we're going to talk about the blind spot, which is referenced here. That's where the two kind of optic nerves come together. But when we're talking about kind of this interpretation. The lens isn't rigid, right? It can perform accommodation by changing shape to focus on near and far images. That's why some people are nearsighted or some people are farsighted. It's that ability to accommodate and perceive text or whatever it is you're looking at from afar or close up. And so how flexible your lens is basically your ability to have 20/20 vision or to have a astigmatism or be nearsighted or farsighted, right? So with that kind of big picture, you can see the basics of the eye in its entirety. We'll get into more of the specifics here and start by talking about the retina. So basically, we see, like we were looking at in that last example, right, the candle was flipped. It was inverted by the lens to the back of the eye, which is where the retina is. So you can see kind of here on the left, I don't know, it's hard to kind of show this stuff without being in person, but we're going to do it. So on the left here, that's like the full eye, and as light is passed to the back of the eye, right, it's inverted, and we take a cross section then of the retina, and we can see what kind of exists within it. We know that the light in the eye triggers photochemical reactions in the rods in the cone at the back of the retina, right, the back of the retina here. We are not going to talk a ton about the ganglion cells and the bipolar cells. Their job really is to start like the chemical neural process that's going to shoot this information into our brain and give our optical nerve and our occipital lobe a chance to like interpret it. So we're not going to talk a ton about that, but we are going to talk about rods and cones because they have a really important role in our vision, and we'll look at that in just a second here. But to give just a brief overview, right, we know the light enters, goes to the rods, chemical reaction activates the bipolar cells, and then the bipolar cells activate the ganglion cells, the axons of which converge to form the optic nerve, right? And so we see the rods and cones like inputting stimulus, sending information to the bipolar cells and into the ganglion cells, which then form our optic nerve, which gets that information directly into our visual cortex in the brain, okay? That's a very basic overview. We are, after we go through all of this, going to watch just like a short video that's going to kind of summarize all of this in a more visually appealing, animated way, so bear with me for right now. Like I said, those ganglion cells converge to create the optic nerve. And you can see, right, the optic nerve in this image right here, right? If we're thinking this is like the rest of the eye is right up here. This is the back of your eye where it kind of connects then through the optic nerve to your brain. There is a blind spot, and you all know this, right? Think like most mug. I don't even know if you guys are taught that anymore. Here, let's draw our little iris. Oops, I didn't like that one. Let's make blue eyes. Ooh, blue eyes. Good old blue eyes. This is fun. Waste of your time, sorry. Most mug in like your driver's ed, you have to like look over your shoulder, check your rear view, check your side mirror, check your over your shoulder. You have to do like 8,000 times to be aware of your blind spot that a car provides. Our eyes also have that. There's an area of missing information in our field of vision known as the blind spot, and this occurs because the eye has no receptor cells at that place where the optic nerve reaches the eye. So like you can imagine if some light hits the eye here, it's going to be transported to the back of the eye here, right? If it comes in here, it's going to be transported and hit the back of the eye here, right? But if it comes straight in and it hits right here, that's your blind spot, okay? Sorry, this isn't perfect, I know. But we know that there aren't receptor cells at the very center back of the eye, and therefore there's a blind spot there. With that said, we are not really ever aware that we have a blind spot, and the reason for that is because our eyes are always moving, right? And the brain fills in that missing information. We're never like really hyper-focused on one specific place. If you are, you might notice that things start to like get out of focus, right? This is an example that we might do if we were in class together. I would put these telephones on the screen and I would say, look specifically at this dot, keep one eye closed and one eye fixed on the dot, and you'll see that one or the other of these telephones are going to disappear. They're going to enter your blind spot and you're not going to be able to actually see or perceive them. But again, you have to have one eye closed, because if the other eye's open, right, your brain is just going to fill in that information. So that's your blind spot. You don't really have to know it, but it's interesting and it gives us further understanding of how our eyeballs work, because they are consistently moving, they are consistently taking in stimuli, and our brain is flipping, inverting those images and filling in the blank spots as we move along. So how that actually happens, right, is through the rods and cones. And you saw this. These were located in the retina at the back of the eyeball, right? We looked at that cross image of the retina, and you saw that there are rods and cones, and we know that those rods and cones are triggering a photochemical reaction. They take in the light and then they send their understanding of what this light is to the bipolar cells and the ganglion cells. For the purpose of prepping for your FRQ and prepping for like what you need to know for this class, you need to know that rods help us see in black and white, and they help with our peripheral view and in the dark. They're about 20 times more common than cones, and cones are what help us see sharp colors and detail in bright light. So our cones are really focused on like the specific stuff that we can see right in front of us. Again, if I'm looking at my kitchen, I see butcher block countertops with different like stripes of wood, all kind of pieced together, different shades of stain, right? Those are my cones. Now there's other stuff going on in my vision. I can see movement in my peripheral vision, right? If I move my hands by the side of my head, you guys, I'm sitting here by myself in my house. And I'm like just doing all these silly actions. But you can envision it. If I move my hands by the side of my head, right, I don't really see my hands, but I see movement, right? Those are my rods picking up that kind of black and white signal. My brain is going to fill in the color, right? And so here you can kind of see what that looks like. Cones are very highly color sensitive. They're very detail sensitive, whereas rods are far less color sensitive and far less detail sensitive. So then moving on to the visual information processing, we know kind of looking here, so this is a kind of a chopped in half version of the bottom half of our brain, so like our eyes and below. If you just were to chop off the top part of your head, you can see kind of the path that this information, the stimulus, moves through, right? The images we see are not made of light. They are made of neural signals, which can be produced even by pressure on the eyeball, right? When you think about, it's all so kind of hypothetical and it's crazy, and I have said this a few times and I'll say it 100 more, like what our brain does when it looks at something is just so incredible. And we know that like this light is then transposed into a neural signal that's sent into the back of the brain. So right, you have light that comes in to your eyeball. It's taken in by the cones and the rods, and it's sent to the bipolar cells, the ganglion cells, transferred into a neural signal that travels down the optic nerve through the thalamus and into the visual cortex, which is where that visual, whatever you're seeing, is being processed, right? Once the neural signals enter the optic nerve, they are sent through the thalamus to the visual cortex, that occipital lobe. I mentioned here that it's not always light that's producing this, but our eyes are so sensitive and our optic nerve is so sensitive, you can actually close your eyes, and if you turn your eye to the left and you press on the right corner of your eyelid, you can see that there is pressure there. You can visually see that like light. It's like when you rub your eyes really hard and you see stars or you see spots, right? That's from the pressure. Or if you like get punched in the head, again, that's the optic nerve translating the stimuli, whatever it might be, pressure or light, into neural impulses that is then interpreted by your occipital lobe. All right, just a couple more before we watch our kind of big picture video. Again, some ganglion cells in the eye signal directly to the visual cortex in response to certain features such as visual patterns, certain edges, lines, or movements in and around the visual cortex of the occipital lobe. Supercells integrate these feature signals to recognize more complex forms such as faces. Basically, this is just a lot of words to say that like different areas of our brain are primed to better interpret different stimuli. So one part of our brain in our occipital lobe is more ready to like recognize faces, and there are parts of our occipital lobe that are more quick to recognize houses or chairs, right? And so you can see where that is on the brain through this image right here. And we also know, and the reason that we know this is because damage to these areas appears to make it difficult to see certain kinds of objects. Specifically, there's a diagnosis called let's see, prosopagnosia. It's more commonly referred to as face blindness. If you have like a stroke or some sort of like brain bleed in the area of your brain that is able to recognize faces, you may end up with face blindness, in which you look at people but you do not recognize their face, right? They can see the features, but they can't integrate them into like an entire, okay, that's Mrs. Ellis's face, or that's whomever's face, right? And that's prosopagnosia. There's a bunch of videos on YouTube about it. You should look it up. It's interesting, but it just goes to show, right, that these specific areas of the brain are in fact primed to be better at interpreting different sorts of visual stimuli. So with that, then we go into our understanding of parallel processing. Parallel processing really refers to the building perceptions out of sensory details processed in different areas of the brain. So really this is just like taking light and turning it into the mental act of seeing, right? Taking all of our perceptions about what is being input into our brain and really like coming together with like an image. So like this example, you're showing your cones are perceiving that color. We're gonna talk about different cues, monocular and binocular cues in a bit, but like you're able to perceive motion, you're able to perceive form and depth, shadows. All of these things are taken into account, and we can say, okay, that is a 3D bird moving from left to right, flying in the air from left to right. Like, so parallel processing is pulling different perceptions in terms of our vision and the stimulus that's actually coming into our eye, turning into those neural impulses and being interpreted by the occipital lobe by way of the thalamus. And we're able to put that together and understand what those objects really are. Here's kind of an overview of what that looks like. We have a scene, right here we have a tiger. Our retinal processing, right? The receptor rods and cones activate the bipolar cells and the ganglion cells, which then sends those neural impulses into the brain. The brain's detector cells respond to specific features. Again, that's that parallel processing, edges, lines, and angles. And you're able to combine those for information about color, form, and movement. And then the last piece of the puzzle is recognition, right? Brain interprets the constructed images based on information from stored images, right? This is a tiger. So with that, we're going to finish up this conversation about like vision as it is like the actual sensation, the process, the interpretation of stimulus by watching this just overview of the visual system that gives you kind of a more graphic visual understanding of what that looks like than I can give you from just talking via this microphone.
Your eyes allow you to see the world around you, but have you ever wondered how they work? First, it's important to know that vision depends on your brain as much as on your eyes. Your eye's main job is to detect patterns of light. Then, they work with your brain to turn those patterns into images. Let's take a closer look. Light rays bounce off an object you are looking at. Let's say the object is a dog. The light reflects off the dog to your eye. Then, the light enters through the outer part of your eye, called the cornea. The cornea is clear like a window. It helps your eye focus the light to make things look sharp and clear. Next, the light rays pass through an opening called the pupil. The pupil is the dark round circle in the colored part of your eye. The colored part is called the iris. It controls how wide the pupil is and how much light can pass into your eye. In bright light, the iris narrows the pupil, reducing the amount of light that enters the eye. In dim light, the iris widens the pupil to let in more light. All of this happens automatically. Behind the iris is the lens of the eye. It helps focus the light coming into your eye so you can see things clearly. The lens flattens so you can see things that are far away, and bends so you can see things up close. When the lens, cornea, and pupil are all working together properly, they will focus light on the back of the eye. That's important because lining the back of the eye is the retina. There are about 130 million tiny cells in the retina that are sensitive to light. When these cells detect light, they turn it into electrical signals. Those signals eventually make their way through the optic nerve, which is like a cable connecting the retina to the brain. The retina helps create a rough image, but it sees the world upside down. It's your brain that turns what you see right side up. Also, when you look at an object, each eye gets a slightly different view of the world. The brain combines those views and makes them into one picture. The brain also adds a lot of details to your vision, so that you can see complex shapes, movement, depth, and a rainbow of colors. And of course, the brain connects your sense of sight to things you already know, so that when you see a dog, you recognize it as a dog, and not a cat or a monkey. And that's how you're able to see. To learn more about how your eyes work, visit nei.nih.gov/kids.
So hopefully that video gave you a little bit better of like a visualization of what that process actually looks like. We're going to finish up our conversation about vision, looking at color, starting with the Young-Helmholtz trichromatic theory, which is the 3-color theory. And basically, according to this theory, there are 3 types of color receptor cones. There are red, green, and blue, like you saw in the video. All the colors we perceive are created by light wave stimulating combinations of these cones, right? So like, think back to like first grade art when they talked about you have your primary colors and you have the ones that they combine to create, right? Our cones are basically doing that. They're saying, okay, I see a little bit of this reddish and a little bit of this bluish, so this must be a purple color. Or be looking at the orange on the left-hand side, and we know that actually this looks a little bit more yellow because it's juxtaposed with that yellow background, but we can see the color of an orange because our eyes are absorbing all the light except for the wavelength that our brain interprets as orange, right? The red and green and blue don't actually refer to the appearance of the cones. They're just the colors to which these 3 cones react. So then taking this a step further, we can look a little bit at color blindness and look at the opponent process theory, which refers to the neural processes of perceiving white as the opposite of perceiving black. Similarly, yellow versus blue and red versus green are opponent processes. So looking at these images over here on the right, you probably would know if you were colorblind at this point, but if you can't see the numbers 45 and 74, you may be colorblind, and that's because in these images, right, green on the top is juxtaposed with that opposite, which is red, and on the bottom, red is juxtaposed with that opposite, which is green, right? Some people say that dogs have black and white vision. In fact, they're just lacking red receptors. So their vision has simpler colors, and it's more like dichromatic more so than monochromatic. With that said, right, we know that our brain processes these colors as opposites. Black is the opposite of white, yellow the opposite of blue, green is the opposite of red, and they're opponent processes within our brain. So we're going to do a quick trick here to show you what that actually means. I want you to keep, what is this triangle from? So I want you to actually just stare real quick, just to give you an example of the opponent process theory. Stare at that dot in the center of the screen. I'll let you stare at it for like 10 seconds. Just stare at it. Don't look away from that dot. Look at the 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 dot. Bottom, bottom. Keep looking. I'm just trying to keep your attention. Keep looking at the dot. Look at the dot. All right. Do you see red and blue on the screen? Because generally speaking, if you stare at that dot long enough, I don't know if that red triangle did anything to change our perception. But this is actually the opponent process for the British flag. So looking at that flag, it had the green X, that would be red. It had the black outline, which would be white, and then it had the yellow kind of fill-in, so that would be blue. So when you are able to put that green and yellow image on the screen and you stare at it, when you take it away and you look at a blank slide, you're going to see red instead of green and blue versus in opposite of yellow. And so that proves the opponent process theory. Wow, I really did not do a good job of explaining that, but you get the drift. So bear with me. We're going to kind of crank through the rest of this PowerPoint slide just because I want you to we're going to talk about kind of these perceptual cues just very briefly, and then you are actually in the activity that I've mentioned a couple of times going to go out and find examples of them. So I just want to give you a basic understanding, and if you need to do a little bit more digging and get some more clarification, you can look at the textbook or like the internet or watch a YouTube video, I don't know. But I'm just going to give you the bare minimum. So with perceptual organization, we know that we have perceptual processes for enabling us to organize perceived colors and lines into objects, right? We are able to group and complete parts into gestalt wholes. We'll talk about that. We'll see figures standing out against backgrounds. We perceive form, motion, and depth. We're able to keep a sense of shape and color constancy despite changes in visual information, right? Think about like if you're looking at something and like the lights turn off, we don't say like, oh, this turned color. We say like, well, the color is the same, the lighting's just different, right? And then lastly here, we're using experience to guide visual interpretation. So let's start by looking at the role of perception, right? Looking at this image to the right, our senses take in the information, but our perceptual processes turn them into a variety of different things depending on who you are and what your experiences are, right? You might see white paper with blue circle dots and a cube floating in front of them, right? You might see white paper with blue circle holes through which you can see a cube. Right? You might see a cube sticking out to the top left or the bottom right. Or you might just see blue dots with some angled lines. I don't see a cube, right? And that's okay. Everybody's going to perceive this differently. And that's what the role of perception is, right? We're looking at an image and we're saying like, what is this? I see the cube in front of those blue dots, but I can also envision the fact that you might see blue with the cube behind those holes, right? With that, then we can kind of go into our understanding of figure ground. This is when we pick out objects and figures that are standing out against a background. So here, what do you see? Right? In most visual scenes, we pick out the objects and figures standing out against a background. So in this first image, we may see two faces looking at each other. But if you take a step back, right, and look at the white, you may see a goblet or like a vase, right? Same thing on the right. We probably will see the man taking a step because that's the figure standing out against a background. But if you look further, right, you see those arrows going in different directions, right? So oftentimes art muddles the ability by giving us two equal choices about which is figure and which is ground. So this is an example of what that looks like. And that's going to bring us into our conversation about gestalts. A gestalt refers to a meaningful pattern configuration forming a whole that is more than the sum of its parts. So when we're talking about gestalt, you will always associate the term gestalt with whole, right? There are three ways, and actually more than three ways that we're going to look at for the purpose of tomorrow's activity, that we group visual information into wholes. These three that are referenced here, we have proximity, continuity, and closure, right? So proximity here on the left, all of these lines are close together, so we group them, right? We usually don't see like 6 individual lines, right? We see that as a group of lines, right? Continuity, that kind of squiggly line, right? We don't see it like here's a part, here's a part, here's a part, here's a part, right? We see it as one continuous line with a straight line through it. And then lastly, looking at closure here, we see those triangles even though there's no like background outlining them. Here, to take that further, similarity is when things are similar. So you see the two taller lines, we would group those together in a gestalt and separate the other two shorter groupings. And then again, that continuity, again, or that closure piece, we see those triangles here, even though they're not outlined. So we're going to dig into each of these specifically here and talk about each of them individually. So the first, Gestalt principle. So that's what we call these. There are going to be, let's see, 1, 2, 3, 4, 5, 6. There's 6 properties of Gestalt principles that we are going to look at. The first one being proximity. And basically that is this idea that we group closer together elements, separating those from further apart elements. So looking at this particular image, right, on the right we see 3 dots close together, and on the left I know my lefts and rights, I swear it. On the left we see 3 dots closer together, and on the right we see 3 individual dots, and our brain automatically groups those 3 that are closer together and looks at the ones on the right as 3 individual dots as opposed to a group of dots. Okay? Law of proximity, we group closer together elements, separating them from those that are farther apart. Intuitive. Similarity then says that we seek differences and similarities in an image and link similar elements, right? So on this far left one, right, this one, right, we're gonna say that there's pink dots, they're in a group, and there are black dots, right? In this one, we're gonna group probably these square dots together, square dots, that's counterintuitive. We see the squares and the circles, right? And then on this last one, we have 1 group, 2 groups, and 3 groups based on color, right? So they're all different shapes, they're all in different orders, but we can group them based on their similarities. The next one then is continuity. We referenced this one a little bit already, but this is the idea that we follow and flow with lines. So here, right, you can see that this is a continuous line, right? But this could also be a continuous line. We don't view it that way because our understanding of continuity, but having the colors gives us insight, and it says, okay, this is one continuous line, right? Here we have a continuous line, and here we have a continuous line. And so based on that continuity, we flow with the lines. And just inherently, looking at these, if all of these were the same color, right, we would probably perceive this as one line and this as one line just based on our Gestalt principle of continuity. The next one we are going to look at is closure. The law of closure means that we prefer completed shapes, and our brain automatically fills in the gaps between elements to perceive a completed image. So we see the whole first. This can be true also, like if you're outside and you're doing your scavenger hunt and there's like a tree here. I don't know, a tree here. And maybe there's like a cloud off in the background, but you can only see part of the cloud. Our brain fills this part in, right? That's closure, okay? And obviously in this instance, like we see a full triangle, we don't just see like a bunch of little lines right here, right? We see a full triangle, we don't just see individual lines. Second to last Gestalt principle is connectedness, right? We group elements linked by other elements. If they are touching, they are grouped, right? So looking at this example, the top right, we're probably going to group these because of similarity, right? But now, if you're looking at this one, all of a sudden they're touching, and we say, okay, these are all groups, right? Even though our blue dots are still the similar dots, that's not the grouping that we here we have these grouped together, right? Same thing here. Here's a group, here's a group, here's a group, here's a group. So based on if they're connected or not, that's how we are going to put them into our defined gestalts. Oops. Last gestalt principle here. Looks like I accidentally drawed on this page. Whatever, drawed. Oh my gosh. It's 4 o'clock. Maybe I need to be done. Drew on this page. The last one here is common fate, and we group elements that are moving in the same direction, right? We have our birds that are flying this way, and therefore they're in a group, and our planes that are flying this way, and therefore they are in a group. That's common fate. Okay. So here's kind of an overview of the Gestalt principles, right? Closure. Even though this G is not connected, our brain fills it in. Proximity, right? Because these are close together, we will group them. Continuity, right? Even though right here the S disappears, we know it continues behind the post. Yeah? Similarity being these two are the same, we know they're both T's, and then figure ground, right? We know that we pull out the A and the L from the background, okay? Just an easy way to kind of remember some of those ideas. So there you have those Gestalt principles. Those are kind of the first cues that we take in terms of perceiving and understanding our surroundings. And we're going to kind of shift our perspective now to binocular versus monocular cues. Binocular, right, by, using both eyes. Just the basic understanding that because humans have two eyes in the front of our heads, this gives us retinal disparity. You will have to understand what retinal disparity is, and that's the fact that two eyes have slightly different views, and the more different the views are, the closer the object must be. So if you put your finger like in front of your nose, and you close one eye, and then you close the other eye, and you see that that finger kind of hops from left to right, right, you know that that finger is very close. Now, if you put your finger as far away as you can in front of your face, right, and you kind of wink your eyes, the finger doesn't move as much, right? So retinal disparity is this idea that when things are closer, they kind of are juxtaposed when you close an eye, versus when they're further away, you don't really see that change in positioning, right? So just kind of think about that idea that binocular cues are always when we're using two eyes and we're integrating like our understanding that because we have those two eyes and we can see kind of the disparity that exists, that's going to tell us how far away something is. That is in juxtaposition to monocular cues, right? Monocular cues are helping us perceive depth from 2D images, right? We only really need one eye to do this, but as we talked about, like when we talked about the blind spot, right, our eyes are constantly moving, they're always taking in information, and so we're able to kind of combine our binocular cues, that retinal disparity, with some of these monocular cues. So like I said, we're going to crank through these pretty quick, just to give you an overview, and then you are going to actually go out into the world and come up with examples of what those actually look like. So the first monocular cue that we are going to talk about is interposition, and this is when an object appears to block the view of another. We assume that the blocking object is in a position between our eyes and the blocked object. Therefore, we can perceive depth, right, because the thing that's doing the blocking is obviously closer than the thing that's being blocked, right? This top image here is showing us kind of how an optical illusion can kind of mess with us. And it shows like the blank space between the trees in front of the horse and the woman. And obviously our brain is like, whoa, 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 that's not right. And we can recognize that's not right because of this idea of interposition. The next monocular cue is going to be relative size. Relative size is that we intuitively know to interpret familiar objects of known size as further away when they appear smaller. So here we're looking at flowers, right? The flowers that are closer to us are big, right? And so we know they're close. And as they get further away, those flowers are smaller and smaller. Funny kind of comic here on the side. I have no depth perception. Is there a cop standing on the corner, or do you have a tiny person in your hair, right? Normal people, obviously this is a joke, but if you didn't have issues with depth perception, you would see like she's larger, so she's up close. The cop is behind her, he's further away, and he is smaller, right? Relative size gives us insight into depth and how far away something is. Next, we have this idea of linear perspective, and this is the idea, if looking at this image, right, the flowers in the distance seem to further away because the rows converge. They come closer and closer together until they meet. Our brain reads this as a sign of distance, right? We see this with like roads or train tracks, which you'll see on the next one actually. But as things that are in straight lines get further and further away, right? Parallel lines seem to converge on the horizon, right? That's our idea of linear line perspective. So I referenced train tracks. We use this linear perspective. These two red lines, right? They look like they're different sizes, but in fact, if you kind of measure them out, they are the same size. They meet the retina as being the same size. However, our perception of distance affects our perception of length, right? So this illusion is actually called the Pranzo illusion, and it was first demonstrated by an Italian psychologist in 1913. The idea is that you place two rods or bars or lodges on the same size screen, but our eyes tend to see one as larger because linear perspective makes its location on the train track seem further away, right? Kind of a fun little trick, fun fact. The next monocular cue that we're going to look at is relative height, right? We tend to perceive the higher part of the scene as farther away, right? So looking at this, the tree, here we go. This tree, right, is higher on this image than this tree, right? And so because it is higher on like the horizon on our visual field, right, that means that it's going to be further away. So relative height. Then we have relative clarity, right? Light from distant objects passes through more atmosphere, therefore they are perceived as hazy and further away than sharp, clear objects. So looking at this image, right, you can see the leaves and the details of these trees here that are kind of like up close, right? But as you get further and further and further and further away, things get hazier and hazier. And that's the idea of relative clarity. And because these things are hazy, we can perceive that they are in fact further away. The next one is texture gradient. Some objects appear closer because they are coarse and more distinct, but gradually become less and less distinct or further away. Now, I just want to clarify that you're not always going to see like all of these in play at once, right? Texture gradient and relative clarity seem pretty similar, right? As it gets further away, it all kind of blends together and gets a little murky, right? But here we're specifically talking about like being able to pick out individual objects in the forefront, things that are closer, and because there's a lot of them and they go off into the distance, right, they begin to kind of mesh and turn into like a single field of purple something instead of individual purple flowers. Or on the bottom here, cracked sand, I actually like a desert image, and as it gets further and further away, you just see it as kind of like chunky ground instead of this like clearly defined cracked earth, right? So you're not going to see all this stuff at the same time, and you're going to pick things out in the activity that you're doing tomorrow to the best of your ability. It will probably not be perfect. Like, we don't live in the mountains, so you're not going to be able to maybe pick out like a hazy mountain scene, but like we're going to do our best here. Again, do note I'm going through these very quickly. That's intentional. I want you to actually like just get an introduction here and then dig into deeper understanding with tomorrow's activity. So the last kind of monocular cue that we're going to talk about is motion parallax. And this can also be referred to as relative motion. And do note when you are doing this example in your activity, you are going to need to take a video to kind of represent this. But this is basically saying that when we are moving, we can tell which objects are further away because it takes them a longer time to pass, right? This particular depth perception cue is often referred to, like I said, as motion parallax, and it is used by many animals that don't have the benefit of monocular cues or binocular cues, right? Those two eyes, being able to see the difference in an object based on how close it is. Relative motion, basically the objects we pass appear to be moving in the opposite direction, and the further away objects don't move as fast. So like this example, this image here, oops, I didn't have my microphone plugged in. Hopefully this helps. I'm gonna double check that. Okay, well, it didn't sound too bad, I don't think. Not perfect, but we'll chug along with the microphone. If you're looking at this particular image, right, if you're sitting on a train or in a car or whatever, right, the things that are really close, the houses that you're driving by, kind of go really quickly and they pass through your visual field, your window, really fast. Whereas the things off in the distance, right, never really seem to move. You never really seem to catch them. And they move a lot slower. And therefore, we know that those things are further away than the houses that are zooming by us, right? That makes sense. We've all experienced. So that was the last of the monocular cues per se. We're also going to talk just briefly about perceptual constancy because it's really important to our understanding. Like I referenced at the beginning, kind of this idea that when the lights turn off, we don't say like, oh, those cabinets completely changed color. We say like, oh, those cabinets are clearly the same color, the lighting just changed. So that's going to be kind of the undertone of this perceptual constancy. And this is really our ability to see objects as appearing the same even under different lighting conditions, at different distances and angles. Perceptual constancy is a top-down process, right? We're referencing from experience the fact that like even though our eyes are taking in like a different stimulus and a different sight, right, our top-down processing says like, well, we know that the cabinets are still white or that they are still like a butcher block top at like a cedar tone, right? So there's a couple of different examples of this. The first one is color constancy, right? This is the ability to see consistency in color when changing illumination. It helps us see all three sides as being yellow because our brain compensates for shading, right? So here, looking at these images, or this particular image, all sides of this cube, we all perceive to be the same color yellow, and we assume that it's the same color yellow. Our top-down processing tells us it is because of shading, right? Because one side isn't in the light doesn't make it less yellow, it just makes the lighting less helpful for us to see it, right? As a result, we interpret the same color blue dots with shades that are not adjusted for shading as being three different colors. So if we look at right, this dot, because it hasn't been adjusted for shading, right, we're like, that's not the same color blue as this dot, right? If it had been adjusted for shading, our top-down processing through color constancy, we'd be like, okay, these are all the same color. Every side of the cube is the same. It just depends on what the lighting looks like. Color constancy. We also have shape constancy, and that's the ability to perceive objects as having a constant shape despite receiving different sensory images. This helps us see the door as rectangle as it opens. Because of this, we may think that the brown shapes on our screen are also rectangles, right? And so like, again, this door here, like the shape of it, what's actually coming into our brain is not in fact a rectangle, right? It's kind of an oblong quadrilateral. My mom's a math teacher. She would be proud that I used the term quadrilateral. Anyway, like we see that, and even though our brain is telling us this is a quadrilateral, it's not necessarily like a perfect rectangle. We're like, but it is. We know it's a door that's opened, and like it's just the angle at which we are seeing this door. It's not the fact that the door actually changed. Again, that top-down processing to reference this. The last example of constancy is going to be size constancy. We have the ability to use distance-related context cues to help us see objects as the same size, even if the image on the retina becomes smaller, right? That's kind of goes hand in hand with the relative, what was it? I forget. Gotta keep them all straight. The relative size monocular cue, like we're able to recognize that just because somebody looks tiny and like we say like that's far away, our top-down processing says like, but we know that if we were up close and personal with that human, they wouldn't be just like a tiny speck, right? They would be in fact a normal sized human. So they did a kind of optical illusion experiment with this idea of size constancy, and it's called the Ames room. It was invented by an American ophthalmologist in 1934. It was designed to manipulate these cues, these distance cues, and try to make two same-sized girls appear very different in size. So I'm going to let us watch this. We have one little final slide after you watch this video with Philip Zimbardo. If you take AP Psych, we're going to talk about Zimbardo. He did the PRISM experiments, the Stanford PRISM experiments. He's a very, very well-known modern psychologist, and he kind of talks us through this Ames room and what it's doing to our perception.
Lights, camera, perception. When you were little and some big kids pushed you around, did you ever fantasize about becoming bigger all of a sudden? Well, I did, and now I can, at the Exploratorium, San Francisco's Science Museum. How did I do it? The easy way, by manipulating your perception. When you first saw me, I appeared to be normal size, because you couldn't see the rest of the room. But as the lights came up and the camera pulled back, your brain performed a new instantaneous analysis. It now saw me as small. It added up all the visual information it had about my size and the size and shape of the room. And then it added in everything it knew from experience about the usual shape of rooms, that they're rectangular and have right angles. So when I walked over here, your brain was convinced I was growing incredibly large because there was no other perceptual explanation, even though it wasn't logical. Let's look at it again, only this time from a different perspective. The room is not rectangular at all. It's totally distorted. There are no right angles anywhere. So that makes this clock, which is large and oval, look the same as that clock, which is small and circular, from your other perspective. The floor slopes upward, and as you can see, the ceiling slopes downward. So I'm not getting larger at all. You're just misperceiving my size because you think that you're the same distance from me when I'm over here as you are from me when I'm over there. But in fact, I'm twice as far away.
So the last piece of the puzzle here is this idea of perceptual adaptation. Over time, if our perceptions are being manipulated, we are able to adapt to that change in perception, right? After our sensory information is distorted, such as by new pair of glasses or by delayed audio on television, humans may at first be disoriented, but can learn to adjust and function. Right on the right here, this guy is a fighter pilot, and he's wearing they're not drunk goggles, but they're similar in that they like shift. I actually have a couple of pairs of these, and if we were in class, I'd have you all wear them. They're super fun. We try to throw balls at each other and give like high fives, and they just kind of mess with they shift it all to the side or flip you upside down. But obviously that's really disorienting at first, but eventually this fighter pilot could learn to fly an airplane wearing these unusual goggles. But at first he can't even find the man's hand to shake, which gives me anxiety because like social distancing and handshaking is a no-go. Anyway, that's the idea of perceptual adaptation. If our senses have been manipulated, if our perceptions have been distorted in some way, we can learn to adjust to that, and our brain does that for us. So there you have it. That is our conversation about vision and perceptual cues. Again, you are going out. My extra will give you a little bit more information on what the activity or assignment is that you're doing tomorrow. Good luck, have fun, get outside, do something moving, and learn about psychology in a fun, different way.